It was established in April 2022. The association was formed, bringing together mycologists with the broad objectives of promoting and disseminating knowledge on different factors.
Today's talk is about phylogenetic analysis. This is mainly for the beginners. If you're a beginner, I'm sure you will benefit from today's talk very much. If you're not a beginner, but you can still get some interesting insights from today's talk, I guess. I hope everybody can listen, hear me clearly. Please give reaction somewhere. If you can hear me clearly, yes, I can. Great, thumbs up is fine. Thank you so much. Thank you so much. Please, hands down. Thank you. I'll switch off my camera and I will start presenting from the slides, using the slides. Rohit, is my first slide visible to you?
Yes, it's visible.
That's everything is fine, I guess. Yeah, and my voice is also fine, I guess.
Yes. It's in full mode and clear.
Yeah, thank you. Thank you so much, ladies and gentlemen, for participating in today's online talk on molecular phylogenetics or phylogenetic analysis for beginners. About me, Rohit has already talked about me. I'm Belle Damodara Shenoy. I'm a principal scientist working at CSIR National Institute of Oceanography Regional Center, Visakhapatnam, which is on the east coast of India in the state of Andhra Pradesh. I'm happy to be the editor-in-chief of the prestigious journal MycoAsia, Journal of Modern Mycology. These are my contact details. If you don't know my contact details, you are most welcome to note down these things. As I have already shared the slides, you can find my contact details in the PDF file I have shared. I have taken most of the content for today's presentation from this book chapter. If you're keen on learning, you can download this book chapter from the internet for free, I guess. So as I told you, today's talk is about phylogenetic analysis. So what is phylogeny? So it's a very commonly used word especially in fungal taxonomy or in any taxonomy for that matter. So basically the fungi is that our genes, our proteins, which are present in our cells actually, they can act as evolutionary chronometers. What is meant by chronometer? Chronometer is something which talks about the time. So if we use the information hidden in the genes and proteins of our cells, then we can get insights about the time, what might have happened in 100 years back, 10,000 years back, 1 million years back. It depends upon the genes or proteins you're going to use. The information will be specific to the genes or gene regions you're going to use it, I guess. So that is the funda of phylogeny. Basically, phylogeny is nothing but the evolutionary relationships of the organisms and genes. So as I told you, this is based on the funda that proteins and genes act as evolutionary watch or evolutionary chronometers, which can help us measure the evolutionary change. So as a researcher, as a beginner, where can you find DNA and protein sequence data? As you might not have prior information, or you might not have done any molecular biology work yet. Where can you get DNA and protein data? You can get these DNA data, protein data from the public databases. What are these public databases? So there is a famous one called NCBI. There's this, and one of the options is NCBI GenBank. You go to NCBI GenBank, you can search for the sequences. There are two ways of searching for gene sequences and protein sequences. So you can do the keyword search. For example, if you are interested in gene sequences of Aspergillus, you go to this option called nucleotide and type Aspergillus. Or if you're keen on a particular gene region, then you say, for example, ITS. Aspergillus, ITS, then you will get the results. It is similar to Google search. If you select one sequence, one Aspergillus sequence, actually, then you can try to get homologous sequences using a tool called BLAST. It's also present in NCBI website. Actually, these are freely available. You can have fun with these tools. Actually, so what can you do with DNA sequence information? You can do many things. One of the things you can do is you can construct phylogenetic trees using DNA or protein sequence data, actually. What is a phylogenetic tree? So like a natural tree, organic tree, phylogenetic tree will have a root, branches, leaves. Okay, I'm going to explain that in coming slides. Basically, a phylogenetic tree is an estimate of the relationships among taxa. Taxa is a very commonly word used in taxonomy. Basically, it means group of taxonomic groups, group of taxonomic categories, actually taxa. And the phylogenetic tree is all about the estimate of the relationships among taxa or the sequences that you are used for representing them and their hypothetical common ancestor. Why hypothetical? Because for many of them, the ancestors might have vanished from this earth. What we have is probably the hypothesis. These could be the common ancestor for a group of fungi that we may be studying. So basically, a phylogenetic tree is an estimate of the relationships among taxa and their hypothetical common ancestors. So phylogenetic trees are nothing new. Even Charles Darwin used the concept of phylogenetic tree. You can see from Charles Darwin's notebook. This is a page from Charles Darwin's notebook where he tries to explain the relationships, different set of organisms and their relationships. So you can see some lines are longer, some lines are shorter. So what is the meaning of that? That's about the Charles Darwin. In the modern day, in biological classification, we have three domains. You have bacteria, you have archaea, eukarya. So this is also a phylogenetic tree. So if you talk about fungi, for macrofungi, the fungi which are easily visible for using our naked eyes, you have a beautiful tree here. So this is also a phylogenetic tree. Actually, it talks about the different groups of macrofungi and their relationships, their evolutionary relationships. For the kingdom fungi, we have a new classification system since 2007 or 6. This is based on the phylogenetic tree using multi-gene sequence information. So this is from Hibbett et al. 2006. A higher level phylogenetic classification of the fungi. So that means phylogenetic trees are widely used in taxonomic research, in mycological research, for that matter, biological research. So I was talking about at the kingdom level, at the species level, how can a phylogenetic tree help you? A phylogenetic tree can help you to identify a species, to accurately, For example, here you have the, penicillium, a fungal culture of penicillium. This is the culture from which the penicillin was originally kind of discovered, accidentally discovered. So this is Fleming's penicillin producing culture. This belongs to the genus Penicillium. And if you want to identify it correctly, what you can do is you can firstly do the morphological study. If you do morphological observations, analysis, you could do this kind of structures. Macroscopically and microscopically. In addition to that, if you extract DNA from this particular bit here, you can sequence a gene region called beta-tubulin or calmodulin or some relevant gene region. Then, based on the gene sequence information or the newly generated sequence, you can get homologous sequence or similar sequences from the public databases, and you can construct a phylogenetic tree. Here is the sequence of Fleming's penicillin producing culture. So, and these are the homologous sequences. Basically, these clustered together, confirming that Fleming's penicillin producing culture is a species of penicillium called Penicillium rubens. So this is the practical use of phylogenetic trees. So that means understanding or mastering the technique of our generating phylogenetic trees is very good. It will help your career if you are into taxonomy especially. So how to construct a phylogenetic tree? So are we doing good? Just give thumbs up if you're doing good so far. Please, great. So people are getting it, great. Let me now go to a little bit complicated part actually. So how you might ask me how to construct a phylogenetic tree. There are basically 4 steps when we get into the phylogenetic tree construction. The first one is assembling a data set. So if you have newly generated sequences from your lab, very good. and you get the sequences, homologous sequences of GenBank using keyword search or using BLAST tool. So you assemble a data set. Once you assemble, you need to align them. So what we have to do multiple sequence alignment. It is not a very simple thing to do. So here, I will talk about multiple sequence alignment when I come to that in the coming slide. Multiple sequence alignment is a very, very critical step in phylogenetic tree construction, where the sequences are aligned in such a way that each column actually represents some kind of evolution actually. So once your multiple sequence alignment of the gene sequences or protein sequences is ready, you can find out the best trees. So there are tree finding methods that I'm going to explain. Once you get trees, so you need to find statistical support for the branching patterns. So statistical support can be understood by a process called by a technique called bootstrapping. So these are the 4 important steps involved in phylogenetic tree construction. First one is assembling a data set. Second one is creating a multiple sequence alignment. Third one is to find out the best trees. The last one is the bootstrapping. These are the 4 major steps. Actually, there could be some minor other steps also that we can discuss later. So before I get into these 4 steps, it is important to identify different parts of a phylogenetic tree. A phylogenetic tree, for example, has these parts. First we have a root here, the oldest ancestor. Then we have branches. These branches connect to sequences, or we'll have branches or lineages. These connect to organisms or to sequences. These goal checker kind of goal things are round things are these are nodes. When they are inside, they are called internal nodes. When they are at the terminal end, actually, or terminal position, they are called terminal nodes. So what are these terminal nodes or the leaves? A, B, C, D. These are basically the sequences that you have used in your analysis. So these are called leaves or terminal nodes. Basically, they represent the taxa or the genes or the population. So what are these? These are not directly used in our analysis. We just make hypotheses about it. So this is an internal node. This represents the hypothetical ancestor of B and C, for example, here. So, and here is the oldest ancestor of all these taxa, actually. Okay, this is called a root. So these are the different parts of a phylogenetic tree. You can start with terminal nodes or leaves, which represent the sequences you use. Then you have internal nodes. Then you have branches. Branches connect these taxa or the nodes. Then you have the oldest ancestor, that is the root. So these are the different parts of it. So nodes, terminal nodes. There are 2 types of nodes: terminal nodes and internal nodes. And terminal nodes represent the extant species, the sequences that you have used in analysis. As I told you, internal nodes represent the hypothetical ancestors. So if these are the terminal nodes, this is an internal node in that way, actually. So branches actually have these. So these are the branches. So you can see these are the lines. So these connect 2 organisms. One could be dead, another could be living. So it actually determines the ancestral relationship. So branching pattern of a tree is called topology. So how does this tree look? That is nothing but topology. It's also used in computer networking, also topology. So we can assign weights to branches or the branch lengths. Some branches are short. For example, this is shorter than this one. So what does it mean? That means here in this situation, the evolution, there is a more amount of evolutionary changes actually. So that is what it means. So what is a rooted tree? I talked about the root. Basically, root is the oldest ancestor. So you can choose to root a tree or not to root a tree. When you root a tree, what happens? It actually gives an idea about the direction of the evolution. For example, this is supposed to be the root. for example. This is the oldest probably known organism. So maybe it is dead, but it is the oldest ancestor. So this could be the root here. And actually, then it talks about the evolution. For example, you have like prokaryotes, then you have eukaryotes, butterflies here, you have fishes, then you have human beings. This is about the rooting of the tree. So rooting is basically to give the direction, to understand the direction of evolution. Actually, how to root a tree? How to root a tree. You can root a tree using multiple species actually. One or more outgroup species. For example, if you're talking about human beings, if you're studying only human beings, then fishes can be an outgroup, actually a root. If you're studying about the human beings and fishes, then the butterflies can be outgroups. If you're studying about butterflies, humans, and fishes, then a lower organism, for example, bacterium can be an outgroup actually. Or when I say outgroup, it is a root actually. So you cannot use any sequence. So there has to be some scientific reason for that actually. Outgroups, basically, it cannot belong to the group of interest. You cannot use as one of the sequences from the humans as info to outgroup, to make it outgroup normal. So it should be phylogenetically separate. So the fishes are phylogenetically separate from human beings. But if you are studying about only about the human beings, probably then it is not a good idea about using bacteria as outgroup. So it should not be very distant also actually. So are we doing fine so far? Please give thumbs up. All right, looks like we're okay. Doing okay. Great. Rohit, is it fine?
Yeah, it's going great, Dr. Shenoy. Yes.
Thank you. So when you focus on, there are two things you should focus on, in a phylogenetic tree. You should focus on the branching pattern, that's topology. Then you should also focus on branch length. Branch length. These are the two things you can focus in a phylogenetic tree. So there is something called a phylogram. So this is a phylogram. Here is a graph. In phylogram, there is topology, there is branching pattern. So okay. Topology is there, and topology is there. That is topology is there. That is the branching pattern is there. Branch lengths are also very clear. So then it's a phylogenetic tree which can give idea about the branching pattern, also about the branch lengths. There is dendrogram. Dendrogram basically talks about the branching pattern. There is no information about the branch lengths. Basically, okay. There are some issues in phylogenetic tree construction that might need your kind attention. What will happen if you add more sequences in a phylogenetic analysis? So if you have 10 sequences, that will result in 3, 4, 4, 5, 9, 4 to 5 distinct topologies, the distinct branching patterns. So many trees. When you increase these sequences to 20, that will result in 82007. These many number of distinct topologies or branching patterns, these many trees. When you add 50 sequences in a phylogenetic analysis, that will lead to 2.75292 into 10 to the power 76 distinct topologies. That means as you increase the number of sequences in your phylogenetic analysis or tree construction, the complexity increases. When the complexity increases, then you would need higher computational power. Sometimes you might need a supercomputer. Okay, so if you add so many sequences, so there is a compromise between number of sequences and the accuracy of the tree you get, because the computational power computational power as it is available to us need not be infinite actually. So then in that case we need to compromise between number of sequences and the accuracy of the analysis. So what will happen if we get so many different topologies, so many number of different trees in a phylogenetic analysis? Then you have to use something called optimality criteria to deal with many distinct topologies. There is something called strict consensus tree. So if you have one, for example, 1,000 trees, then you will take an average of everything. Average of average of certain things actually, then the resulting tree will contain exactly those groups that present in all the source trees. This is something called a strict average kind of thing, what you call strict consensus tree. In other kind of tree, majority rule consensus tree, in that kind of averaging, you would get a tree that contains exactly those groups that are contained in more than 50% of the source trees. I was talking about multiple sequence alignment. Okay, so this is how a multiple sequence alignment looks like actually. So every column is a character, and every here you have, for example, let me give some interesting example here. Okay, last but one, this his full column is a character, and you have C and T there. So these are character states. Okay, this is how we actually make sense of evolution. Multiple sequence actually estimates the homologous nucleotides between the sequences. Homologous, they basically test the hypothesis of homology. So more important, this is more important than the tree building methods. I'm going to talk about tree building methods soon. If you find some of the regions are not aligned properly, you may exclude them from the analysis. Remember, you might get a multiple sequence alignment, but if your multiple sequence alignment is not good enough, as they say in computer science, garbage in is garbage out. Okay, there are two major tree building methods. First one is first tree building method. Tree building method is a method by which you actually generate trees, or using the data set. So these are two. There are two tree building methods. First one is distance matrix methods. Second one is discrete data methods. I'm going to explain that. In distance matrix method, you have a multiple sequence alignment. First, the multiple sequence alignment is converted into a data matrix, the program. So then the program uses the data matrix to generate the tree. So this is the distance matrix method. In this discrete data methods, for example, parsimony or maximum likelihood, the multiple sequence alignment is used as it is to generate the tree. Here, the data matrix part is not there. So these are some of the complicated stuff. So as I was telling, there are two major tree building methods. First one is distance matrix method, where the data matrix part is included. And then there you have discrete data method, where the data matrix component is not there. Examples for the distance matrix methods include neighbor joining, UPGMA. These come under distance matrix method. The parsimony, maximum likelihood, Bayesian methods, these come under discrete data method, which is also known as character-based methods. So there are tree types like UPGMA. I don't think many people are using it nowadays. Then there is something called neighbor joining method. It's a very fast method. Is used for the neighbor joining method is used for the analysis of the large data sets, widely used for constructing phylogenetic trees from distance data. Actually. Then you have maximum parsimony. Basically, maximum parsimony. The idea of parsimony is stinginess. In Hindi, what you call kanjus. So in this philosophy of maximum parsimony is that evolution is very stingy. Evolution doesn't happen in a very expensive way. So there are minimal changes happens in evolution. So that is how the evolution works, very slow and very conducive type. So that is a school of thought behind maximum parsimony. It is also used to it was used quite widely previously. Neighbor joining method results in one tree, but maximum parsimony can result in multiple trees actually. Okay, but then how do you decide? Then you take the average actually. That program does itself actually. Then you have what we call probabilistic methods of phylogenetic inference actually. Basically, here you involve lots of statistics and the assumption of assumption involving models about the mechanisms of DNA substitution. For this, you need to understand the evolutionary biology better actually. Maximum likelihood is one of the probabilistic methods, very powerful. Basically, what happens in maximum likelihood, it is based on the idea of maximizing the probability of observing the data under a set of hypotheses. This is one of the most successful approaches in statistical inferences. Then you have what you call Bayesian analysis. This is based on the Bayes formula. I'm not very good statistics, but better to have some understanding of it if you are very keen on doing the Bayesian analysis. So this is then you have called MCMC approach, Marco chain Monte Carlo approach for approach to derive phylogenetic hypothesis. Then these are all the tree building methods. Okay. Then once you have trees, you need to get the statistical supports. How do you know which branch has got good support? So what we do is like bootstrapping actually. Bootstrapping is what happens in bootstrapping. It is a very simple thing. A program does it automatically if you give the instruction to it. So, in reality, what happens, the program takes random subsamples, build trees from the subsamples, then they calculate the frequency of occurrence of different parts of the tree. So basically, bootstrapping is the test of the phylogenetic accuracy of the tree. Normally, bootstrap values represented here in the percentage, 70% or higher are likely to indicate the reliable groupings. So once you get the tree with the bootstrap, trees with bootstrap values, you might like to edit them. You can use some programs. So here, some things to remember. Branch lengths are drawn to scale. You need to mention the scale somewhere. If you're talking about the branch lengths, bootstrap value should be displayed as percentages. Okay, and you have Bayesian values. You can also mention that, and you can choose to root the tree. If you root the tree, it will give an idea about the direction of the evolution. So I'll speak for next 5 minutes, talking about some of the important softwares. First one is MEGA. So MEGA software is a very popular one. The full form of MEGA is Molecular Evolutionary Genetics Analysis. So this is the recent version of MEGA. I think it's MEGA 12. It's in the development stage. So you can do many things. Basically, what is the purpose of MEGA software? You can do sequence alignment. You can align your sequences in MEGA. You can construct phylogenetic trees. Then you can do model analysis, evolutionary analysis, other stuff. So basically, these are the features. Of MEGA. It's very friendly, user-friendly graphical interface. You can see here there are so many buttons, and you can use your mouse effectively and get the things done. User-friendly graphical interface. You can do neighbor joining. You can do maximum likelihood. You can do maximum parsimony. You can do statistical tests like Bootstrap in MEGA. This is a very good program for beginners and intermediate users. You can download the software for free from the internet. Second one is a little bit complicated for the what we call molecular clock analysis. So the software is BEAST. It's called Bayesian Evolutionary Analysis by Sampling Trees. It results in Bayesian tree. Okay, it does Bayesian analysis, but physically you can input the data about the fossils. Fossil data information will be incorporated in the analysis, that will result in molecular clock. So for a clock, you need to do calibration, right? So in BEAST, you can do phylogenetic analysis using Bayesian method and incorporating fossil data. It's a very powerful software, but probably very difficult to learn also for a beginner. But if you are keen on learning phylogenetic analysis at the advanced level, you should master this software, and it is freely available. And what you can do, as I told you, you can do Bayesian inference of phylogenies and divergent times. You can do coalescent method models, relaxed molecular clocks, and it integrates molecular and temporal. Temporal data means fossil data and other stuff. This is best for dating evolutionary events and analyzing fungal divergence. This BEAST can be downloaded from the website for free. Then you have very powerful, powerful software, Mr. Base. This Mr. Base software looks like this. I'm in my computer, which is Microsoft operating system based. This is for the Bayesian inference of phylogeny. It uses MCMC methods. It estimates tree topology. It basically results in Bayesian trees with branch lengths, and you can use model parameters. This is widely used for reliable Bayesian phylogenetic reconstruction, and this is freely available from this website. Then you have, for maximum likelihood method, you can do the maximum likelihood method in Mega software, but some people do not accept probably certain limitations present in Mega software with respect to likelihood analysis, because likelihood analysis, ML analysis, is computationally intensive. So maybe Mega has compromised on some certain aspects of it. So to overcome that, the researchers are using software. It's basically a web server actually called RAXML, Randomized Accelerated Maximum Likelihood Method. What you have to just upload your data set in certain format, and you have to specify certain parameters. So you will get maximum likelihood tree. And it is kind of a cloud based, it's not a really cloud, but web server based. The computational power aspect will be dealt with, because all the analysis will be done on the server, which is not your computer. Okay, so basically the purpose of RAXML is to construct phylogenetic tree using maximum likelihood method. This is very fast, scalable, ideal for large data sets, and it also does bootstrap analysis. And as they say, this is the best for large and complex data sets. Any data sets, not only fungal data sets. It is freely available, and this you can use it for free actually. Then you call IQ3. This is kind of the interface you have. This is for the fast and accurate phylogenetic tree construction using maximum likelihood. One more software for maximum likelihood analysis. If you see recent papers in fungal taxonomy, two methods are very widely used: maximum likelihood and Bayesian actually. Okay? So these two softwares are very important, All these platforms are very important. XML, IQ3 are very important actually, for advanced users actually. So what are the features of IQ3? IQ3 is for the automated model selection. When you do the ML analysis, it does ultra fast bootstrapping, and it does many more complicated stuff. This is for the cutting edge, what they claim, the cutting edge maximum likelihood tree analysis. This is freely available. You can download from this website. So this used to be my favorite software, but it is very difficult to use. POP, phylogenetic analysis using parsimony. It involves scripting and all. So if you're good with the scripting, you try to use this POP. Probably there are better softwares, but you might still use it for the parsimony analysis and some bit of other stuff actually. Then sometimes Mega may not align large data sets for alignments. Then you can use these web server called MAP. So here, if you have a data set of 500 sequences, 4,000 sequences, you upload it. Within a short period of time, you will get multiple sequence alignment. Also, as I told you, the purpose of MAFT is to create multiple sequence alignment. It's very fast, it's accurate, because multiple sequence alignment, the sequence alignment is a critical step in phylogenetic tree construction. If you have so many sequences in your data set, then it might computationally intensive or challenging. So then the servers like MAFT can help you. Actually, it will result in fast output, accurate output, and it will be as these are suitable for large data sets actually, and the output is high quality. There is one more alignment platform, Clustal Omega. You can use the Clustal Omega. So this is also for fast and scalable for large fungal data sets. If you want to edit a tree, then you can use a software called Fig Tree. It's a very beautiful software. You can do many things. You might see many beautiful trees on in our mycology papers, taxonomy papers, phylogenetic papers, and fig trees widely used for editing the trees, not cutting the branches and all, for beautifying the tree. Okay, mainly the aesthetic purpose. So basically, purpose is the improve the visual. I mean, it's for the visualization and annotation of phylogenetic trees. You will get highly customizable outputs. You can edit the tree layouts. You will get publication ready phylogenetic trees after this. If you use Fig Tree. So I came across this Philo suite. This has got many options. So you can do alignment. You can do phylogeny using different softwares. It's like an environment actually. It's a desktop platform. It's freely available. I guess you can try it. You can do it's integrated software for phylogenetic and evolutionary analysis. You can do alignment, trimming, model testing, tree construction. It supports MAF, XML, Mr. Base. It's very good. Seems very good. So, and it is best for streamlined phylogenetic analysis. It seems like you have got many things in one place. So maybe not a bad idea to try it out actually. And there is one software called Trim AL. So purpose of this software is to trim the alignment files. So it removes poorly aligned regions and gaps for better phylogenetic inference. This is best for pre-processing alignment data. As you can see, this is a command line thing. So you might need some practice before you start using it. BioEdit, it used to be my favorite. When I was doing my PhD, it was my favorite, one of the favorite software. So it does many of these things. Actually, it does manual alignment, correction, sequence analysis. May not be a great one if you want to convert files. So this is also a very good option, BioEdit actually. All right. With this, I conclude my presentation. Thank you very much for listening to me. And if you have any questions, please raise your hand. What do you?
Thank you, Dr. Shenoy, for the talk. I think we'll open this session for question and answers.
Badrina, do you have any question? If you have any questions, please raise your hand. Arinda, okay, let me allow him. Okay. Yes, Arinda.
Hello, sir.
Yes.
Good morning, sir. Sir, what is the concept of bootstrapping you're talking about? Can you explain that part a little?
Bootstrap is basically to test the statistical robustness of the tree that you get. So if you see some 100 in the branch on a branch, that means out of 100 times you will get that kind of branching. Certain on some places you have bootstrap value of 75. Okay, that means out of 100 times, 25 times you may not get this kind of branching. So if you get bootstrap values less than 70, that means less than 30% of times the branching pattern may not be the same thing what you see actually. So basically bootstrapping actually gives test the phylogenetic statistical significance of all the branching actually. So what it does, program does is takes random subsamples from the random subsample data sets. It creates, if you say 100 replicates or 1000 replicates, it will takes 1000, for example, 1000 random subsamples. From each subsample, it will generate a phylogenetic tree. Then it will makes an average of the statistical support for each of these branches. This is how it does, Arinda. And there is a beautiful video on YouTube about the post-trap analysis. I hope I answered your question.
Thank you, sir. Yes, sir.
Rajeshri. Rajeshri, please ask your question.
I want to know, there are different kind of tree building methods. Which one is a particularly applicable for a particular research or particular scenario? How to decide that, or how is that decided? Thank you, sir.
It depends upon your reviewers. Actually. If the reviewer is happy with neighbor joining tree, fine. If the reviewer says the neighbor joining is not enough for your hypothesis or your paper, then they might ask you to do maximum likelihood Bayesian analysis. Basically, you start with neighbor joining analysis. Okay, then you get kind of a tree. Then you can test. You can try other methods, see whether you are getting similar trees. But better to try different methods. Then you decide which one to show in the paper. But basically, try 2-3 methods. I would say neighbor joining, maximum likelihood. If they want, 2 methods should be okay to start with neighbor joining and maximum likelihood. If the reviewer asks for the Bayesian, then you do the Bayesian. Rajeshri.
Thank you, sir.
Thank you. Afifa, please. Afifa, Kainat. Afifa, can you hear me? Dr. Ashok? Dr. Ashok?
Hello, sir.
Dr. Ashok? Yeah, May. Okay, sorry. Yeah, May. Who is this, Afifa? Hello, who is
Yes,
this?
it's Afifa.
Yeah, Afifa, please ask your question. Yeah.
Yes, sir. Sir, it was wonderful presentation. I have a question regarding motifs that we used to cut before moving toward the phylogenetic tree. That motifs are specific, like if we use for ITS or LSU region. And I have a question that the same motifs should be cut in other phylogenetic tree, like if we are using other markers like ITS or except ITS or LSU, we use REF1, REF2, or TAF region or some other regions. So we used to cut same motifs or some other kind of motifs.
I mean, it is not a bad idea to try new markers, okay? But taxonomic analysis, if your genus of interest has 55 or 100 species, somebody has to sequence, all that particular new gene region in all those all those species, right? So priori information, if it is available, then it is easy to try out a new gene marker. Otherwise, better to stick to the established gene markers, Afifa.
Right, sir. Thank you so much.
Thank you. May Ashok Pariyappan.
Sir, good afternoon, sir.
Hi.
Thank you for your informative presentation, sir. It is so good and informative also.
Thank you,
Sir, I
ma'am.
have one question, sir. This means those are all the software is also applicable for bacterial alignment, bacterial sequence alignment, sir.
See, phylogenetic analysis can be universal. You can use the same softwares for bacteria, animals, for human studies also. These softwares are not specific for fungi. Okay, this is the phylogenetic analysis for beginners. So these beginners could be from any field. As I am a mycologist, I give examples from fungi.
Okay. Another one question also I have, sir. If I construct the tree, what is the scale? It is good, it is correct, or it is wrongly I constructed. What is the scale, sir? How can I identify it? Is any idea, sir?
Only the reviewers can tell whether it is good enough or not, or your teacher can tell you. There is no scale for such things. Okay, the correctness of it. That's why now, as I told you, garbage in is garbage out. You can get a tree from any multiple sequence alignment. Okay, but there has to be some taxonomic insight when you create the multiple sequence alignment. Okay.
Okay, thank
So
you so much, sir.
there is no scale, but only thing is there is a statistical support for the branching. If the if the tree is overall good enough, then the branching pattern and the branch support or the statistical support, booster support, can actually convince the peers actually.
Okay, thank you so much, sir.
Thank you. Rajesh Krishnan. Rajesh Krishnan. Rajesh.
Am I audible, sir?
Yes, you are audible now. Please ask your question.
Yes, sir. Thanks for your webinar. It was very much informative, and I could learn good number of new softwares also. My query is related to sequencing, sir, because I'm a beginner for phylogeny. Maybe through this, I will take it up. While going for ITS sequencing, I'm facing a problem of low E value. That is, it is not coming 0.0. But when coming to 16s, I am able to generate a sequence with a proper E value after trimming. So from there, when I'm taking it for mega software, I'm finding it difficult to generate a phylogeny. So can you just advise me like how to trim or how to take up the sequence for ITS, and especially I'm using ITS and also this TEF, translation elongation factor for fungi.
So you meant to say when you do the blast, you don't get the good quality homologous sequences? You mean to say that?
Yes, yes, sir.
Either that group is poorly studied, or your sequence could be something new. Okay, so you should be happy if it is the case of if it is the latter case. You should be unhappy if the former case.
Yes, sir.
If group is poorly studied, so that means somebody has to study it. Actually, there is no other way.
Because around 83 to 90% I'm getting the similarity, but in the mid at least some 60 to 70 base pair I'm unable to get it even after doing multiple times resequencing. So how to proceed further for phylogeny?
What is the name of the fungus? Is it a fungus?
Sir, pardon, sir?
Is it a fungus?
Yes, sir. It is a fungus. I'm working on this, sir. WAM. Arbuscular mycorrhiza.
But you consult the experts in the field. They might have a solution for this problem, because I'm not sure how many people are doing the taxonomy of WAM. Okay, so it depends upon how many people are generating the sequence. Yeah, so you talk to Dr. Sridhar or Evelyn from Northeast. Okay, thank you.
Right, sir. Thanks for your time, sir. Thank you.
Thank you. Thank you. Thank you. Madam Sabitra, can you hear me? Please ask your question. Sabitra, madam.
I think she's not able to hear us. Or...
Yeah. Can you hear me, doctor?
Yes.
Good morning.
Good morning. I can hear you, Madam Sabitra. Please ask your question.
Okay, mine is, I think, very simple. I haven't started with this analysis part, but I have enough ITS data. So I was just wondering, when sir was presenting on the number of specimen we use, so you said that the more number we use, it becomes more complex, right?
Yes.
Yeah, so what could be the standard? Because for example, I have like around 50 plus Amanita ITS data, so I'm yet to analyze that. So I was just thinking, what should I decide? Like, should you go for 10 per analysis or like the 50 at one go?
No, you can sequence even 500 actually. It depends upon the diversity of the sequences. If the sequence is quite diverse, then it becomes computational very challenging actually. Okay, if you're studying only one species or one genus, for example, even 500 sequences should not be a problem because the diversity may not be that high actually. Okay.
Oh, okay. So the number becomes complex if you are not I mean, if you don't have the idea what genre that is. For example,
Yeah.
mine is I have like around 50 for Rusula, 50 for Amanita, so I can analyze 50 numbers at one go.
No, you can sequence 500. If in neighbor joining, you can sequence 500, 1000 also. You will get a tree. You will get a tree.
Okay.
You can use some web servers like you can use some of these servers like Max, RaxML, and these servers can accommodate more than 1000 sequences. Don't worry about that. I was just trying to impress upon the fact that many things happen when you start adding more sequences. Yeah, conceptually, even practically also.
Okay, thank you so much. Thank you so much.
Dr. Rajesh, please.
Thank you, Damodar. Thank you, everybody. It's nice and quite interesting to hear all of you. And there are a number of questions that are being raised for the time being, and I think there is quite keen interest on many people there, perhaps to work on phylogeny. There has been a few questions before, and I would like possibly if organizers allow me to comment a little bit, maybe, or maybe share my slide. I've got a few slides that I can share. It's up to you if time is available, right? Maybe just like 5 or 6 minutes just to give a resume. I know Damodar has done quite well, but maybe I will try to use some of my slides just to answer maybe one or question, or I can just do it right now, which is fine. Up to you.
You can share your slides, please. Thank you.
Okay. Can you see my slide?
It's coming up. It's coming up. Yeah, I can see. Just go to the full presentation mode, please. Thank you.
Okay. But I'll be very quick. Very quick. It's just like a lecture which I gave before, so I'll just be very quick.
We can't see. We can't see the full presentation mode. Can you present from the PDF, please?
It's okay. Maybe
try now. Yeah.
Is that okay now?
No. Just present from the PDF. Make a PDF file if you have.
No, I don't have a PDF file.
Okay, it looks good now. Yeah, thank you.
This one better. Okay.
Yeah, please.
Okay, guys. I'll be very quick and just maybe give you some insights about whenever you go into the world of phylogenetics, what are the important considerations? I understand Dr. Belle has already given everything, but I have 5 minutes with me, so I thought about sharing my experience a little bit. Let's keep this one. So when we talk about DNA sequences, you've got to understand that we can use it in a number of scenarios, especially as Dr. Shenoy said, we can use it in taxonomy. Now, where exactly in taxonomy, we've got to understand what level we want to investigate potential relationships among species. Is it at the class level, at the family level, at the general level, or are we just trying to look at the species? Sometimes there are some genus that has got a number of species in those genera, and therefore we can work below the species level and look a little bit at DNA and their differences below species, for example, at variety level, at pathotypes level, as well as subspecies. When we look into our alignment, there are things that we need to look into. For instance, When we check our nucleotides, we've got to see where are the changes, where are the substitutions that are occurring, and what types of mutations are there. Sometimes we've got single nucleotide polymorphism, and it's something very important whenever we talk about alignment. Whenever you go ahead and extract DNA, usually this is the basic stuff that we use. We go for cultures, we extract the DNA. Sometimes, under most circumstances, you don't have, let's say, the cultures are the starting material. And when it comes to fungi, we can try to pick the fruiting body and extract DNA right from it. So there are different possibilities if you don't have cultures to extract DNA. An overview of the steps, I think Dr. Shenoy has already explained. Usually with your DNA, you go across, your DNA, trying to understand what gene you're using, and then you basically go for sequencing. With that sequences, you get an alignment, and from those alignments, what you can basically do, you can use different types of softwares in order to refer phylogenetic relationships among groups of species. As you can see over here, with your alignment, you feed it somewhere like a black box, and it gives you a tree. And from that tree, you try to infer which species is more closely related to another species. Now, coming back to what you said about, using DNA, we've got to be very careful because we're spending a lot of money and time as well. So we've got to do very careful planning. And what's important in here about how many specimens we have, and do we have sufficient cultures for one particular study? And this is very important. That is the amount of sequences that you have, the amount of cultures that you have, and what type of specimens you are looking at. Again, when it comes to your phylogenetic tree, if you want to have good support, if you want to have, let's say, a good tree, there is something called your taxonomic sampling. And it is one of the most important determinant if you want to have accurate phylogenetic estimation. So the question arises as to whether you need a few taxa and more genes, or you need more taxa and less genes. So now it's up to you to decide, and you've got to know the history of that particular genus you're working on, and that particular species that you're working on, how many taxa are available in GenBank, how many genes have been sequenced before, and where are you going with your study. And this is, you need to get a whole picture first. It's not just about going and sequencing the IMTECH and do one particular tree. So you've got to understand the history behind it a little bit. So I think one lady asked a question about how many groups should I include, how many taxons should I include. You need to understand first what is your in-group taxa. As you can see over here, you've got your in-group taxa, those that you are studying. Now, let's say if you're working on one particular genus, and there are 5 or 10 species in that genus. So, of course, you need to go and include all of them and perform your analysis. If a particular family you're working on has got 5 to 10 genera, you just need to include all of them. But now you might end up in a difficulty if there are diversity of species, for example, more than 400 known species, more than 200 species, then sometimes you've got to be a little bit selective. But again, this will have something to do with your in-group. What representatives are you working on, and which one is important? So obviously, it depends also upon your experimental design. If you're working, for example, on Colototricum, and if you feel that you're working on one particular complex, then obviously you need to focus mostly on that particular complex when it comes to your taxon sampling. So again, when it comes to taxon sampling, okay, what do you want to test, which is very important? What are your objectives of your study? Do you want to include the type species? Of course. Don't just go ahead and repeat previous studies. you've got to come up with something new always whenever you do phylogeny. So as Dr. Bell said, if there is a need, you need to seek assistance and consult the experts. Our group, I think Damodar has mentioned that before, it relates to something about how you polarize your characters in your tree, and it helps us a little bit to infer speciation event as well, especially how our characters evolving across a particular tree. There was something on, choice of genes. Yes, that's the question, which was quite interesting. So which genes do we use? So one thing that is very important is that it depends upon which taxa you are investigating on. And you've got to understand what is the amount of divergence in that particular genes that you are going to use. It also depends at which taxonomic level you're investigating your phylogeny. Some people work at high-level taxonomy. For instance, if you're working at relationships within, let's say, a particular order, a particular family, then you will not be tempted to use genes that are evolving very fast. You will use, for instance, the ITS and the PF and beta tubulin to possibly try to find relationship within closely related species, because those sequences are evolving quite fast. But at higher taxonomic level, you tend to use the slow evolving genes. Which genes do we use again? Well, there are a lot of genes nowadays, protein coding genes, RDNA genes. Now it's up to you, as I said before, to understand, the context, especially the taxonomic context in which you're working. Maybe I think that would be it for me. Okay, maybe I can capitalize on this one. So some important consideration in phylogenetics, first of all, you need to get the sequences right, especially your sequences that you're working on. And I think one audience raised an issue about, how do I ascertain, for example, if I'm doing a blast search and so on. So you've got to understand blast search, it works on a similarity indices. It does not really reflect biological relationships and evolutionary relationships. You mentioned that you've got a disparity in the sequences available in GeneBank and in your sequences, which is perfectly right. It's good. It might be possible, we worked that before, especially with environmental sequences in metagenomics, and we've got some kind of novel lineages, novel clades popping up. So you might think that possibly your sequences are erroneous, but no. Once you check that your sequences are right, okay, it just have something to do that the number of sequences available in GeneBank does not really reflect the number of species available in nature. So obviously, if you're going to blast, you're going to blast it with those minimum of species available in GeneBank. So now it's up to you how you go ahead and interpret your data. That would be it for me. I don't want to take long, guys. Okay, thank you.
Thank you, Dr. Jeevan. Maybe you may please stop sharing so that we can see your face, please.
Okay.
I mean, this has been a teaser for your upcoming talk at MycoAsia, Dr. Jeevan. Thank you very much for that.
Thank you.
Yeah, I think, ladies and gentlemen, if you didn't know, Dr. Rajesh Jeevan was one of the PhD advisors at the University of Hong Kong during my PhD during 2003 to 2007, and he's at the University of Mauritius now. And as I suggested just a while back, this is just a teaser for his upcoming talk. Dr. Jeevan, thank you very much for that.
My pleasure, Dan.
Yeah, we will be in touch. Thank you so much. Swagata Sarkar, please ask your question now. Swagata Sarkar. Yes, Chikanalla. Yes, Chikanalla.
Hello, sir. I'm audible.
Yeah, Sugata, please ask your question.
Yeah. Very good afternoon, sir. And thanks for your very good presentation. And sir, my question is simple. Like you have listed down some softwares for phylogenetic tree. So can you tell me which software will be suitable for microbiome analysis?
Oh, these softwares are not suitable for microbiome analysis. This is mainly for the culture-based sequences obtained from the cultures, actually, not for the culture-independent studies.
Okay. These are suitable only for the culture dependence only.
Mostly,
Okay,
yes.
no,
Yeah.
mostly.
Yeah.
Okay.
Because when you do microbiome analysis, you'll be dealing with millions of sequences, at least thousands, 1 lakh sequences, right? Short fragments. So this software may not be suitable for all those sorts. There are specific software, specialized softwares for microbiome analysis. These are not the ones for the microbiome analysis.
Okay, sir. Okay. Thank you, sir.
Thank you. Eshi Kanala, can you hear me? Still a viewer? Okay. Any more questions, ladies and gentlemen? Rajashri, ask your question.
Hello, sir. I have saw a past presentation regarding use of Genius Prime software to combine two sequences. Is there any such feature available in any of the free software such as Mega? Thank you, sir.
I think it's a kind of a paid tool, actually. Not sure there is any open access thing, actually. You need to do some Google search and find out. As far as my information, there are not many tools which are freely available for that purpose. We need to do some.
Thank you, sir.
Thank you, Rajashri. Any more questions? Yes, Chika, are you there? Over to you, Dr. Rohit Verma, please.
Thank you, Dr. Shenoy. So if there are no more questions, and at the end, I think I'll thank Dr. Shenoy for an excellent talk. I think you have nicely explained about the rooted, non-rooted trees in the beginning, then NCBI gene bank, MLSA, maximum parsimony, NJ, ML, basin analysis, everything you covered. You also explained about the bootstrapping for the beginners and all the major softwares being used for in the culture dependent analysis, and some of the for increasing the aesthetic like picture and other things also very important for a good publication. So thank you for covering that also. I think the number of questions itself shows the interest and curiosity the talk has generated among the beginners. And at the end, I think please, once again, we encourage all the participants to be the members of the Association of Fungal Biologists of India, and also submit your manuscripts and research works at MycoAsia, which is an open access, which has a fast publication, which has no fees involved, good expert editorial board, and has a global reach. We also thank Dr. Rajesh Jeevan for the additional information and add-on brief presentation he shared with the participants. And at one stage, we had 206 participants. So thank you all the participants for joining, and several talks are lined up. I think keep in touch for the updates. Thank you so much.
Yeah, I would like to thank Dr. Rohit for managing the show today. You have been awesome, and I am thankful to Dr. Deshmukh. I am thankful to all the members in the audience. You have been awesome. Thank you very much. Keep supporting us. Jai Hind.